Hello everyone! Today we're diving deep into the fascinating world of muscle physiology. Have you ever wondered how your muscles generate force even when you're just sitting still? Or what exactly happens inside your muscles when you lift something heavy? By the end of this video, you'll have a solid understanding of how muscle contraction works and the factors that influence muscle strength and movement. As always, if you're aiming to boost your knowledge and excel in your studies, don't forget to check out King of the Curve for top-notch learning resources. Let's start by talking about muscle tone and tension. Even when you're sitting perfectly still, maybe meditating or just relaxing, your muscles aren't completely at rest. They're slightly contracted to stabilize your joints and bones. This continuous, mild contraction is known as muscle tone, and it's essential for maintaining posture and readiness for movement. Now, imagine picking up a heavy backpack filled with books. The force your muscles generate increases significantly beyond normal muscle tone. This active pulling force exerted by your muscles during contraction is called muscle tension. To understand how muscles contract, let's look inside a single muscle cell, or muscle fiber. Inside each muscle fiber is the sarcoplasm, which is like the cytoplasm in other cells but specialized for muscle function. The sarcoplasm is packed with long, cylindrical structures called myofibrils. Think of them as the muscle's machinery. Each myofibril is made up of repeating units called sarcomeres, which are the fundamental units responsible for muscle contraction. Sarcomeres are composed of two main types of protein filaments, thin filaments made of actin and thick filaments made of myosin. These filaments are arranged in a precise, overlapping pattern that's crucial for the muscle's ability to contract. Let's delve deeper into the sarcomeres architecture. At the center is the M line, where the thick myosin filaments anchor. The ends of the sarcomere are marked by Z discs, where the thin actin filaments attach. Each thick filament is surrounded by thin filaments, creating an overlapping pattern essential for muscle contraction. Within the sarcomere, there are distinct regions. The I band contains only thin actin filaments and appears light under a microscope. The A band encompasses the entire length of the thick myosin filaments and appears dark. This band includes areas where thick and thin filaments overlap. The H zone is a slightly lighter area within the A band where there's only thick filament with no overlap of thin filaments. So, how does muscle contraction actually happen? It involves a process known as the sliding filament mechanism. When a muscle contracts, the thick myosin filaments pull the thin actin filaments toward the center of the sarcomere, called the M line. This action brings the Z discs closer together, effectively shortening the sarcomere and contracting the muscle. Interestingly, the A band remains the same length because the thick filaments don't change length. However, the I band and the H zone become narrower as the overlap between actin and myosin increases. At full contraction, the I band and H zone may nearly disappear due to the extensive overlap. Now, let's meet the key players at the molecular level. First, we have the myosin filaments. Each thick filament is made up of many myosin molecules. You can think of a myosin molecule as having a long tail and two protruding heads. These myosin heads are crucial because they're the parts that actually interact with actin during contraction. On the other side, we have the actin filaments. The thin filaments look like two strands of pearls twisted around each other. Each pearl is a G-actin protein, and each one has a specific site where a myosin head can attach. The interaction between actin and myosin during muscle contraction is called the cross-bridge cycle, and it involves several steps. First, the myosin head needs to be energized. It does this by binding an ATP molecule and hydrolyzing it into ADP and a phosphate ion. This reaction releases energy, which cocks the myosin head into a high-energy position, similar to pulling back the string of a bow. Next, the energized myosin head binds to the exposed active site on the actin filament, forming a cross-bridge. The release of ADP and phosphate from the myosin head then triggers the power stroke, where the myosin head pivots and pulls the actin filament toward the M line. After the power stroke, a new ATP molecule binds to the myosin head, causing it to detach from actin. The myosin head then hydrolyzes the new ATP, recocking and readying itself for another cycle. This process repeats as long as calcium ions and ATP are available, resulting in muscle contraction. Several factors determine how much force a muscle can generate. The size of the muscle fibers plays a significant role. Larger muscle fibers have more myofibrils, meaning they contain more actin and myosin filaments. This allows for more cross bridges to form and results in a stronger contraction. The number of active muscle fibers is also crucial. Recruiting more muscle fibers increases the overall force, much like having more people pulling on a rope in a tug of war. The frequency of stimulation affects contraction force as well. When a muscle is stimulated more frequently, calcium ions build up in the sarcoplasm because there's not enough time for them to be pumped back into the sarcoplasmic reticulum. This accumulation enhances the force of contraction. The length of the sarcomere, known as the length-tension relationship, is another important factor. There's an optimal sarcomere length where the overlap between actin and myosin filaments maximizes force. If the sarcomere is too stretched or too compressed, the force generated decreases because the overlap is not optimal. Finally, the velocity of muscle shortening influences force production. The speed at which a muscle shortens affects the force it can produce. Slower movements allow more cross bridges to form, resulting in greater force. Conversely, rapid contractions result in less force because there's less time for cross bridge formation. Let's consider how the length of a muscle affects its ability to generate force. Take your biceps muscle, for example. When your arm is slightly bent, the sarcomeres are at an optimal length, allowing maximum overlap between actin and myosin. This position enables the strongest contraction. If you flex your arm fully, the sarcomeres become compressed. The actin filaments may overlap excessively, and myosin heads might run into the Z discs, reducing force production. On the other hand, if you extend your arm completely, the sarcomeres stretch, decreasing the overlap between actin and myosin. Fewer cross bridges can form, leading to a weaker contraction. The force-velocity relationship describes how the speed of muscle contraction affects the force generated. When you lift a heavy object slowly, your muscles generate more force because there's ample time for many cross bridges to form. In contrast, rapid contractions, like throwing a light ball, result in less force because there's less time for cross bridge formation. Think of it like pulling a rope in tug of war. Pulling slowly allows more teammates to grip the rope and contribute force. If you pull too quickly, fewer teammates can grab on in time. Muscle contractions come in two main types. Isotonic contractions involve the muscle changing length while the tension remains relatively constant. This includes concentric contractions, where the muscle shortens while generating force, like lifting a dumbbell. It also includes eccentric contractions, where the muscle lengthens under tension, such as lowering the dumbbell back down. In contrast, isometric contractions occur when the muscle length doesn't change, but tension increases. An example is holding a plank position. Your muscles are working hard to keep you stable, but there's no visible movement. Let's see these concepts in everyday activities. When you're climbing stairs, your leg muscles contract and shorten to lift your body upward. This is an isotonic concentric contraction. If you try pushing a heavy bookshelf that doesn't budge, your muscles generate tension without changing length. This is an isometric contraction. Calcium ions and ATP are essential for muscle contraction. When an action potential reaches the muscle fiber, it triggers the release of calcium ions from the sarcoplasmic reticulum. Calcium binds to a protein called troponin, causing it to change shape. This shift moves tropomyosin away from the binding sites on actin, allowing myosin heads to attach. ATP provides the energy for the myosin heads to perform the power stroke. It's also necessary for detaching the myosin heads from actin after the power stroke. Without ATP, muscles would remain in a contracted state, which is what happens during rigor mortis. So, how does a muscle relax after contraction? It starts with the removal of calcium ions. Calcium ions are actively pumped back into the sarcoplasmic reticulum using ATP. This decrease in calcium concentration causes troponin and tropomyosin to return to their original positions, covering the binding sites on actin. Without calcium, new cross bridges can't form. Meanwhile, existing cross bridges detach when ATP binds to the myosin heads. This allows the myosin heads to release from the actin filaments. With no cross bridges and the binding sites on actin covered, the muscle fiber relaxes and returns to its resting state, ready for the next contraction. Let's recap what we've explored today. First, we learned about muscle tone and tension, how even at rest your muscles maintain a slight contraction to stabilize your joints. We then looked inside a muscle fiber and discovered the sarcomere, the engine of muscle contraction. We explored the sarcomere structure, understanding how the arrangement of actin and myosin filaments is essential for muscle function. We delved into the cross-bridge cycle, where myosin heads bind to actin, powered by ATP and regulated by calcium. We discussed the factors that influence the force of muscle contraction, including muscle size, the number of active fibers, stimulation frequency, sarcomere length, and contraction velocity. We differentiated between isotonic and isometric contractions, seeing how muscles can change length or maintain the same length while generating force. We highlighted the crucial roles of calcium and ATP in muscle contraction and relaxation. Finally, we saw how muscles relax, resetting the system for the next contraction. And there you have it, a comprehensive look at how your muscles work, whether you're lifting heavy objects or simply sitting still. Understanding these mechanisms not only deepens your appreciation for the human body, but also provides valuable knowledge for fields like medicine, sports science, and physiology. If you found this deep dive interesting and want to explore more topics with engaging visuals and interactive content, be sure to check out King of the Curve. Our platform offers a wealth of resources to help you master complex concepts and stay ahead in your studies. Don't forget to like this video, subscribe to our channel, and hit that notification bell so you never miss an update. Thanks for joining us on this journey through muscle physiology, and we'll see you in the next video.